In this video, we're going to talk about amino acid oxidation and the urea cycle. So when you take in proteins from your diet, they're obviously broken down into amino acids. And the first thing that happens is those amino acids are used to make more proteins. Because there are some amino acids, like these essential amino acids here, that we don't have the ability to make within our cells, and so we get them from our diet. So the first priority then is to take those amino acids and use them again to make proteins. Now, if we have an excess of protein in our diet, if we have an excess of amino acids, then they can be used for the biosynthesis of other things. Okay, they can be used for the biosynthesis of nucleotides and biological amines and so on and so forth. Now, if there's even more than amino acids than we need, so we don't need any biosynthesis of nucleotides or anything like that, then those amino acids need to be stripped of their amino groups to be excreted, and the resulting carbon skeletons, called alpha-keto acids, then can be converted into forms that can be oxidized. So what we're going to look at then is some of the reactions of some amino acids, of how we strip them of those amino groups and to turn them into these alpha-keto acids, and see what kind of familiar intermediates we turn these amino acids into. In addition, we're going to look closely here at the urea cycle and see how is it that we take these amino groups from amino acids and safely convert them into urea, which is a safe way to excrete nitrogen waste. So first, let's look at the metabolic fates of amino groups. And this reaction right here is kind of the central reaction for ridding the cell of amino groups. And what happens is we transfer those amino groups from amino acids. So here we have some general amino acid and to alpha-ketoglutarate. So this is alpha-ketoglutarate is the common kind of middleman for excreting amino groups. It's the one that kind of carries it, the first acceptor from the amino acids from dietary or muscle proteins. So that amino group is transferred to alpha-ketoglutarate, and the way these amino transferases work is they always convert a carbonyl to an amino group. So they get rid of this carbonyl, right? And this is, let's take a look, alpha-ketoglutarate here is an intermediate of the citric acid cycle. And when we remove this double bonded oxygen and replace it with an amino group, what we're doing is we're giving it an alpha amino group, an alpha carboxyl group, and then there's our R group, right? That's why we have glutamate. That's what this amino acid is here. But again, glutamate in this particular context is just acting as a carrier of amino groups that will eventually be dumped off to liver cells. Now, when this amino acid here, when it dumps off its amino group to alpha-ketoglutarate via the action of this aminotransferase, it becomes, right, when you remove, just the opposite that we did here, when you remove an amino group, you replace it with a carbonyl. This is called an alpha-keto acid, and this can be converted into citric acid cycle intermediates, pyruvate, or acetyl-CoA, depending upon what amino acid we started with. Now, there are some special things that happen within the muscle. So here's that reaction again that we just got finished looking at. Muscle protein is broken down into amino acids. Those amino acids dump off their amino group to alpha-ketoglutarate to make glutamate. But to ship those amino groups to the liver, instead of glutamate going directly into the blood, glutamate takes that amino group that it just got from all those amino acids and it dumps it off to pyruvate. And when you look closely at the structure of pyruvate and you replace that carbonyl oxygen with an amino group, you'll notice it turns into alanine. So now we have pyruvate carrying the amino groups from all those proteins that have been broken down, all of the amino acids, and that alanine goes into the blood, it goes into the liver, and then it dumps that amino group right back off to alpha-ketoglutarate to become glutamate. That glutamate then in the liver, that's going to donate those amino groups to the production of urea for safe excretion. Now, when alanine donates its amino group off, right, with the same enzyme, just in the reverse direction, it again becomes pyruvate. Through gluconeogenesis, it becomes glucose, which returns to the blood and back to the muscle to become pyruvate. So you've got this beautiful cycle here in which the net effect is to move amino groups from the muscle to the liver where it can be safely excreted as urea. Let's take a close look at this. Let's take a close look at getting the amino groups to the liver. Now, this is the top half of an important figure you should really know well. And what we have here then is the reactions necessary to dump amino groups off from the various surrounding tissues and blood to the liver. Now, I boxed everything here in different colors, and I hope it doesn't confuse matters more than it helps. But here's alanine. We already discussed this. This is coming from muscle. Here are amino acids coming from elsewhere. And when they get into the cytosol of the liver cells, they are involved in this aminotransferase reaction, where the alanine, for example, dumps off its amino groups to alpha-ketoglutarate to become glutamate. That alanine itself then becomes pyruvate again. These amino acids do the same thing. They dump their amino groups onto alpha-ketoglutarate to become glutamate and themselves become an alpha-keto acid. Now, what exactly alpha-keto acid they become? Well, it depends upon which amino acid they were. For example, if they were alanine, that alpha-keto acid they become would be pyruvate. So as they enter the cytosol of liver cells, all of these amino acids, whether it's alanine or any of the other amino acids, they're going to dump their amino group off to alpha-ketoglutarate to become glutamate, and into the matrix that glutamate goes. Now, that glutamate is going to dump its amino groups off to one of two places, and it depends upon the relative concentrations of things and regulation and so on and so forth. Glutamate can dump its amino group off to oxaloacetate, becoming aspartate. Take a look. Here we have that carbonyl oxygen. When you replace it with an amino group, suddenly you have what very distinctly looks like an amino acid. There's an alpha carboxyl, alpha amino, and there is the R group. So it's just one reaction that separates an intermediate of the citric acid cycle, oxaloacetate, into a amino acid. And then that releases alpha-ketoglutarate. Optionally, glutamate will donate that amino group to a bicarbonate molecule to make something called carbamyl phosphate. And these are going to be the two sources of amino groups for urea. Urea is essentially two amino groups connected by a carbon. There's the source of the carbon, bicarbonate in carbamyl phosphate. Here's one amino group from carbamyl phosphate. Here's the other amino group from aspartate. Right? So now keep that in mind, and also keep in mind that we're in using aspartate as a carrier of amino groups here, we're depleting citric acid cycle intermediates. So we got to make sure we find a way of replenishing any of the oxaloacetate that we take out as we are disposing amino groups. And we'll get to get back to that in a moment. All right, so the urea cycle. Now we're looking at the second half of that big figure. So here is aspartate, which is essentially oxaloacetate with an amino group added to it, coming out of the matrix into the cytosol, and it's going to be donating itself to the urea cycle. Here's carbamoyl phosphate, also donating itself to the urea cycle. The urea cycle is simple. It looks complex, but it is simple. Now, you're not required to know the structure, memorize the structure of any of these intermediates, but you should know the 4 steps. Okay. You also should know, here's urea right here. It is a carbonyl, right? There's the double oxygen carbon with 2 amino groups added to it. What's the source of this carbon? Carbamoyl phosphate. What's the source of this amino group? Carbamoyl phosphate. What's the source of the second amino group? Right here, aspartate. Okay? And then understand the way this cycle works. Ornithine picks up a carbamoyl phosphate to become citrulline. Citrulline picks up aspartate to become arginosuccinate. Fumarate drops off arginosuccinate, giving us arginine. And then urea drops off arginine to recycle our ornithine. And so stoichiometrically, the only thing that is happening is we are losing 2 amino groups and a carbon. 2 amino groups and a carbon go in, and 2 amino groups and a carbon go out. The aspartate that went in comes out as fumarate and goes right back into the mitochondrial to replace that oxaloacetate that was used to hold the amino group. Okay? And then urea is excreted in your urine. So in that case, think very carefully about ornithine. Ornithine is playing the same role as 2 other molecules we have previously discussed. So this is a strategy that cells use quite commonly. We have this organic molecule which is acting as nothing more than a holder or a carrier. It carries the carbamoyl phosphate. It carries the aspartate. Then stuff happens, fumarate drops off, urea drops off, and then we recycle it. It carries another one, carries another aspartate. Stuff happens, and it's recycled. Okay, so think about that. What other 2 molecules is ornithine similar to? Okay, now let's just remind ourselves, as we've used oxaloacetate to grab an amino group from the matrix, right? So basically traveling as aspartate, we are depleting the citric acid cycle. But, right, here we have fumarate that is released. That fumarate is going to go right back in and replace that oxaloacetate. So this cycle is not going to lead to any net loss of citric acid cycle intermediates. In fact, let's take a closer look at what's called the TheKrebs Bicycle. So here is our oxaloacetate, and here it's carrying the amino group that it gained from glutamate, which glutamate itself carried all those amino groups from everything coming in from the extra cellular tissues. So now there is essentially, aspartate is essentially oxaloacetate carrying an amino group. It goes into the cytoplasm. It donates itself to the urea cycle, and then eventually fumarate is released from the urea cycle. That fumarate gets converted back into malate, okay, via that enzyme. And then malate, remember, there is a transport protein for malate, so malate can move back into the matrix and become oxaloacetate. So again, the net effect of that particular cycle is simply to move an amino group from the matrix to the cytoplasm for disposal via urea. Also, take a look at this. Now, when oxaloacetate is converted to aspartate and aspartate moves here, we also have that malate aspartate shuttle where aspartate can be converted back to oxaloacetate here, then to malate, and then back in. And in doing this, right, we have a way to shuttle electrons via NADH here to NADH here in the mitochondrial matrix. And so all of these pathways are interconnected, right? Instead of each one being something independent of each other, they all have various connections. They all reuse common molecules over and over and over again. They use strategies over and over and over again, like this ornithine being used as simply a carrier molecule. So again, I want you to understand these reactions, these basic aminotransferase reactions that occur. And I want you to remember what is the source of the 2 nitrogens and the carbon of urea. And be able to explain steps 1 and 2 and 3 and 4. Don't worry about explaining 2A and 2B. Just explain it as one step. All right, well, let's take a look now at these alpha-keto acids. Basically, what happens, let's look at some specific amino acids and see what happens when you remove their amino group and you send them down a specific set of reactions. What familiar intermediates do they become? Well, when you look at it, it's really only 7 products. All 20 amino acids can be broken down into one of these 7 products, or some of them into a couple of these products. Acetyl-CoA, acetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, and pyruvate. Now, the ones that are converted to acetyl-CoA and acetoacetyl-CoA, those cannot be converted into glucose. They are not glucogenic, and so we call them ketogenic. They can instead be converted into ketone bodies. The other ones, since they can be converted into intermediates of the citric acid cycle, they therefore can be involved in gluconeogenesis and the production of glucose, and so we call them glucogenic. You don't have to memorize all of these, but here are the ones you do have to memorize. Look how easy it is to turn an amino acid into a familiar intermediate. We have serine, one reaction becomes pyruvate. Alanine, we've already seen this. One reaction becomes pyruvate. Glutamine. Glutamine, one reaction brings it to glutamate. Okay? And then glutamate, one reaction brings it to alpha-ketoglutarate. Asparagine and aspartate, one reaction necessary to convert asparagine into aspartate, and then yet another reaction to convert aspartate into oxaloacetate. In fact, this is a reaction we've already seen before. So these are the amino acids. Now, you don't have to memorize the enzymes that catalyze each one of these steps, except for the basic idea of what an aminotransferase does. But you do need to know the connection. Asparagine to aspartate to oxaloacetate, glutamine to glutamate to alpha-ketoglutarate. You should memorize these.